Good evening. Yesterday we started the topic bacteria, where is a prokaryote having no definite nucleus and no mitochondria, no chloroplast, no endoplasmic reticulum, and no Golgi bodies. And then it is omnipresent. Everywhere you can find bacteria, and the bacterial cell having this outer slime layer, and then plasma membrane, and inside you have the cell organelles and RNA, and very important, the mesosomes. Those are found in the center. They are the infoldings of plasma membrane. And now let us see the reproduction in bacteria. The reproduction is of three types: vegetative reproduction, asexual reproduction, and sexual reproduction. Regarding the vegetative reproduction, there you can find the binary fission. In this is also called a fission or binary fission, where there is no involvement of chromosomes. Here the nuclear material itself becomes two, and at the center of the mesosome, generally I told you already, the mesosomes are found in the center, and from the center they help in the formation of the cell plate during this binary fission, and then a constriction prepared or formed in the center, and the constriction proceeds and form two cells. So the separation of the cells, it is called a centripetal division here. Centripetal, that is from the periphery to the center, and later two daughter cells are formed. It is a quite a common type of binary fission, and the total it takes 20 minutes. So within 20 minutes, one cell becomes two cells, and then the formation of endospores. During unfavorable conditions, the bacterium, especially the bacillus type of bacteria, secretes a thick wall around it and take rest. And at the time, the nuclear material, including the plasma membrane, etc., goes to one side, and a thick, very thick wall around it. You can find here the layers of the endospore. And then whenever the conditions are favorable, the protoplast inside the bacterium comes out, breaking the wall, and comes out. And I told you already, one bacterium gives one bacterium only, so we cannot consider as a reproduction. It is a tied over the unfavorable conditions only. So generally it is known as, the endospore is known as a bacterial cell inside another one, a spore inside the bacterial cell. Next, you see the sexual reproduction. Actually, the sexual reproduction means the fusion of male and female gametes and formation of the zygote. It is diploid. It undergoes reduction division and form haploid individuals. But here, it is not a true sexual reproduction, and only a recombination of genetic material or a transfer of genetic material from one bacterial cell to another bacterial cell. So this recombination takes place in three types. One is conjugation, another is transduction, another is transformation. Now let us see the conjugation. This conjugation process was first reported by Lederberg and Tatum in 1946 in E. coli bacteria, Escherichia coli bacteria. And cell-to-cell union occurs between two bacterial cells. Cell-to-cell union occurs between two bacterial cells. The genetic material of one bacterial cell, here what happened, when two bacteria come together and the donor bacteria having the sex pili, recipient bacteria does not have the sex pili. So with the help of the sex pili, two bacterial cells come together, and then the genetic material of one bacterial cell, actually this genetic material in the F factor or F plus factor or fertility factor, that is found only in the male bacterium, that is in the plasmid, and it is attached with another cell. A copy of this DNA passes through the conjugation canal from one cell to another cell. So now what happened? Later the recipient have the characteristics of the donor. This donor is male. And then what happened? The recipient becomes male. So the female cell becomes male cell through this conjugation. So this is a very interesting type of reproduction. The thing is, transfer of genetic material from one cell to another cell. So the another cell, a recipient cell, having the characters of this donor one. And next one, the process here you can see clearly. Two bacteria come together, one with having the sex pili and other one do not have the sex pili. And now the plasmid, here the red color one is a plasmid, and after joining of the two, a conjugation canal is formed, and then a copy of the DNA passes from one cell to the another cell. Here you can have, and then immediately they are separated and form into the two cells are separated, and this recipient having the characteristics of the donor cell. The next process, the transduction. Here what happened? This was first discovered by Ginder and Liederberg, 1952, in Salmonella typhimurium. In this type, the genetic material is transferred from one bacterium to another bacterium by a bacteriophage. We have already studied the life cycle of bacteriophage. In this bacteriophage, through the bacteria, the thing is, transfer of genetic material or a phase-mediated genetic transfer. The genetic material is transferred from one bacterium to another bacterium through the bacteriophage. You know that bacteriophages are of two types, the virulent lytic and temperate lysogenic. So in the first one, the virulent or lytic phase, what happened? The bacteriophage infects the bacterium, and the phage DNA enter into the bacteria, and this order to prepare its own phage particles, they assemble together and release. When release, what happens in this meanwhile? Some of the bits of the bacterial genome or bacterial DNA incorporate with the phage DNA and form the bacteriophage. After releasing, this bacteriophage infects another bacterium. It carries the genetic material of the previous bacterium to the next stage bacterium. That is one type. And the second one, the lysogenic. Here what happened? The incorporation of this phage DNA with the DNA of the bacterium, prophase is formed. And then after some time, their association breaks, lysis takes place. Again, the bacteriophages are released. Bacteriophages are released. So in the same way, these released bacteriophages, the released bacteriophages or some newly formed phages have the pieces of the bacterial DNA. The newly formed phages attack another bacterial cell. The DNA of the first bacterium is transferred to second bacterium and acquires new characters. This is also I told you, this is a transfer of genetic material from one bacterium to another bacteria through these phases. And the next one, this transduction process, as I told you already, here you have two types of transductions you can have, and the infection of the bacteriophage takes place and incorporation takes place, form a lysogenic one, and then this association breaks and formation of the new phage DNA and cell lysis, release of the phages. The released phages having the genome of the bacteriophage and also the parts of this bacterium also. Next, the transformation. This was first studied by Griffith in 1928. in pneumococcus or pneumonia causing bacteria, Diplococcus pneumoniae. This bacteria causes pneumonia disease. It leads to the death of the individual, whether a human being or animal. In this bacterium, there are two strains. One is non-virulent R type. Non-virulent means never causes the disease. This type of bacterium never causes the disease. The second one is the virulent type, that is called the S type, and it causes disease. So you see, one strain, the same Diplococcus pneumonia, one strain causes the disease, that is virulent disease. And the second one, non-virulent, it does not contain the capacity to give disease. So now, in this method, the genetic material of one bacterial cell goes to another bacterial cell in a medium by some unknown mechanism. The mechanism not known, but the genetic material of one bacterium goes to another bacterium in a medium. By this one strain of bacterium is converted into another one. One bacteria, for example, a R type is converted into S type of bacteria. In this above experiment, the non-virulent R type is converted into S type of bacteria. And here, Griffith could not guess the transforming principle. He has conducted the experiment, but he could not understand how one bacteria turns to another bacteria is one process. Another thing is, what is the thing that is transferred from one bacteria to another bacteria, whether it is a cell wall material or whether it is a DNA. But later, A.V.V.S, McCarthy and MacLeod in 1944, they conducted this experiment and later found out that the transforming principle is DNA. The DNA is transferring from one bacteria to another bacteria so that the bacteria having the characters of the poisonous one. So this experiment, you can see now, very interesting experiment. The virulent bacteria, we have this culture of the virulent bacteria, and it is virulent bacteria is injected to a mice dead. And then they conducted post-mortem and observed the body cells and their cells containing the virulent bacteria. In the second experiment, non-virulent bacteria injected to the mice survived, and absence of bacteria. They never find any bacteria in this blood. And again, what happened? The heat killed virulent bacteria. They are killing the virulent bacteria because they cause disease, but it is heated, and then injected to mice. Mice survived. Mice is survived. So no effect, though it is a virulent because of the heat killed. And then in its blood, absence of bacteria. No bacteria is found. No bacteria is found. And then the next experiment they conducted is heat killed the virulent bacteria and non-virulent bacteria. Heat killed virulent bacteria and non-virulent bacteria. Actually, the virulent bacteria, heat killed virulent bacteria cannot give the disease, and non-virulent bacteria cannot give the disease. They combined both and injected to the mice. It's so surprising, the mice dead. So they observed again, and they take the samples of this blood and study. In its blood, they found virulent bacteria. Very interesting. Separately, non-virulent cannot give the disease, and heat killed cannot give the disease, but they mix both, it gives the disease. So through the medium, the DNA passes from one cell to another cell, so the R type becomes S type and causes the disease and kill the mice. So this is the transfer version of genetic material from one animal to another animal or one cell to another cell through the medium. Next, let us see the economic importance of bacteria. the economic bodies is a large account. There you have to remember a number of bacteria. So in this, we can divide this economic importance into two types. One is harmful aspect, another is beneficial aspects. In the harmful aspects, the first one, deterioration of materials. Most of the materials you can see, the paper or the cordage, ropes, rubber, cork, etc. materials, etc. are deteriorated after some time. Just you are having a book, after 10 or 20 years, if you open, you have some patches and the paper will be not pleasant. So this is by many bacteria like Mycobacterium, Pseudomonas. They destroy the cork materials, rubber materials, cordage or ropes and paper, etc. Next, water pollution. So generally, it is majority of the bacteria and some algae, they spoil the water, drinking water. These bacteria like Vibrio cholerae, Salmonella typhi, they spoil the drinking water. Not only spoiling the drinking water, if you drink those water, we get those diseases like cholera and typhoid, etc. by the water contamination. The next, spoilage of food. Spoilage of food. Generally, it is a burning problem where most of the roadside hotels, etc., they keep materials, the yesterday's material, today's materials yesterday, and also I heard some papers that most of the people dead, those they consume some biryani, etc., in a particular hotel because of this bacterial infection only. So, Pseudomonas, Escherichia, Micrococcus, Salmonella, Staphylococcus, Clostridium, etc. spoil the foodstuffs, especially the carbohydrate products, and cause food poisoning. Botulism is a very serious food poisoning caused by the Clostridium. It leads to the death only. Regarding the diseases, they contain this many bacteria cause diseases in plants and animals and human beings. The several plant diseases show some symptoms like galls. Galls means bulging portions or bulging of some portions of the leaves. Canker means the leaves become black. Leaf spot, you can find some spots on the leaf, some yellow spots on the leaf. And blight means just like fired paper. Soft rot, that is rotten like, etc., are some of the important plant diseases. The list is given below. So regarding the diseases in plants, the citrus canker disease is caused by Xanthomonas citrae, and it will affect not on the leaves but also on the fruits also. The cankers are found on the fruits so that it reduces the yield of the fruit and also the juice content of the fruit. And second one, there is bacterial blight of rice, that is Voriaceae. The total inflorescence is become blight and yielding is reduced. And angular leaf spot of cotton, that effect on the cotton leaves, and their bacteria is completely eaten this chlorophyll, and the production will be reduced. Regarding the fire blight of pears caused by the Pseudomonas solanacearum. So these are the diseases: citrus canker, bacterial blight of rice, angular leaf spot of cotton, fire blight of pears. Next, soft rot of carrot. Generally, you can notice in the markets that most of the carrot may be decayed. That is called the soft rot by the Erwinia carotivora. And brown rot of potato, Pseudomonas solanacearum. Ring rot of potato. Ring rot of potato, that's if you cut a potato, inside you can find a black ring is there, so that the weight of the potato will be reduced, caused by the Corynebacterium sepadonicum. The crown gall of plants, galls of plants, Agrobacterium tumefaciens. So here, soft rot of carrot, brown rot of potato, ring rot of potato, crown gall of plants, These are the plant diseases, some of the plant diseases. And next we will see the diseases in animals. In animals, the serious disease is anthrax of sheep caused by the Bacillus anthracis, and brucellosis of cattle caused by Brucella apodus, and brucellosis of sheep, goat, etc., Brucella melgensis, and TB in cattle, dog, and parrot, that is Mycobacterium tuberculosis. So these cause the diseases: anthracis of sheep, brucellosis of cattle, brucellosis of sheep and goats, TB in cattle. These are the diseases of animals. Diseases in human beings: cholera, Vibrio cholerae. This is water contaminated. We can have this cholera disease, as I told you already. These are the spoiling the water, Vibrio cholera. So this is a water contaminating disease. And the typhoid by Salmonella typhi, TB, Mycobacterium tuberculosis, and diphtheria, Corynebacterium diphtheriae, and pneumonia fever, just to know in this transformation, Diplococcus pneumoniae, pneumonia causing bacteria. So cholera, typhoid, TB, diphtheria, and pneumonia are some of the diseases. And next we have some more diseases of the animals. Human beings, the plague, Yersinia pestis. Sometimes you have a petis also, and tetanus, Clostridium tetani, whooping cough, Haemophilus pertussis, and dysentery, Shigella dysenteriae, and leprosy by Mycobacterium leprae. So some other diseases are plague, tetanus, whooping cough, dysentery, and leprosy. Next we have the regarding these harmful aspects, biological warfare. This is a very dangerous thing. So you shoot, you can kill some people, and bombing also a group of people. But whereas the biological war, just if you take a capsule of, for example, this botulism bacteria, anthrax bacteria, cholera bacteria, tuberculosis, etc., just take in a capsule and just you throw in a particular area, the whole area will be suffered with the disease and they will die. So it is a strong weapon to be used against enemy. Regarding the beneficial aspects, antibiotics are excellent things. So the first antibiotic we have known is penicillin by Alexander Fleming, obtained from the penicillium, that is a fungi. But here these antibiotics, majority of the antibiotics are Waxman. Streptomyces is the second one in the antibiotic list. What is an antibiotic? It is a chemical substance which produce from one microorganism, it kill the effect of another microorganism. The following are some of the antibiotics obtained from bacteria. A list is given. Please follow slowly, because I have given 12 antibiotics and also the bacteria, name of the bacteria from which they are obtained. Sertraline from Bacillus subtilis. Polymyxin, sorry, here the spelling I have you change it here is poly, P-O-L-Y, polymyxin, X-I-N, polymyxin, that is Bacillus polymyxa, and Serixin, Bacillus ceres, Biosirine from Bacillus ceres, and Bacitracin, that is Bacillus licheniformis, and licheniformis is also another antibiotic. It is obtained from the same bacterium, Bacillus licheniformis. And streptomycin, I told you already, this is prepared by Waxman. Streptomycin from Streptomyces gracilis. Chloromycetin and Streptomyces venezuelae. And terramycin Streptomyces remosus, and Aromycin Streptomyces aerophaciens, Erythromycin Streptomyces erythris, Neomycin and Streptomyces radiae. So, it's a list of antibiotics. You can note down, it is for interest. Sertraline, polymyxin, dirixin, biocinin, bacitracin, licheniformis, streptomycin, chloromycin, terramycin, aromycin, erythromycin, neomycin. So next we have the beneficial aspects in a dairy industry. Without bacteria, we cannot get curd. So the change of milk into curd by the bacteria only. Milk products like curd, butter, cheese, etc. are obtained by the action of bacteria. Lactic acid bacteria, for example, Streptococcus lactis, are employed to convert milk sugar lactose into lactic acid. So this is the formation of the milk to curd process. Sewage disposal. This is a very tedious problem in developed areas. What is sewage? Sewage is nothing but the domestic and industrial liquid waste. So daily we have an enormous amount of liquid waste, so that you have to recycle or you have to convert into another products. The organic material of the sewage is decomposed by many saprophytic bacteria. So the sewage disposal process is a combination process of algae and bacteria. So generally what happens, the sewage may be given into several compartments of sewage, and then a number of algae which rapidly give the photosynthesis and you give oxygen. That oxygen is used by the bacteria to decompose the waste materials. So this is sewage disposal, a joint program of algae and the bacteria. So the organic matter of the sewage is decomposed by many saprophytic bacteria. Next, decomposition of a plant and animal remains. Every day or every minute, number of organisms, a number of plants and a number of animals die, and they may be thrown to some other places, so that you can feel that the smell in the next day or after 2 or 3 days. Who will clean? The bacteria. Saprophytic bacteria cause decay and decomposition of the dead bodies of the plants and animals. So they release gases to the atmosphere and salts to the soil, and so these bacteria are known as nature's scavengers. So previously the scavengers used to clean the fecal matters, but now it is stopped. But now these bacteria are known as the natural scavengers because they used to clean the dead bodies, etc. by the decomposition. Another very important thing is the soil fertility. So bacteria are important agents in maintaining soil fertility by the following ways. One is nitrogen fixation, ammonification, and nitrification. By these processes, the bacteria fix nitrogen in the soil. Regarding this nitrogen fixation, they are carried out by the atmospheric bacteria. The asymbiotic bacteria, like first separated into symbiotic and asymbiotic bacteria. Asymbiotic bacteria like Clostridium, Azotobacter, Chromacium, Sporophyllum, Rhodophyllum, etc. fix atmospheric nitrogen into nitrogenous substances and fix in the soil. And the symbiotic nitrogen fixing bacteria like Rhizobium, they fix the nitrogenous materials in the root nodules of legume plants. Root nodules of legume plants. So they supply to the plants. These nitrogen fixing bacteria having the NIF genes, So they have an attachment between a particular rhizobium bacteria and the legume plant. These nitrogen fixing bacteria, rhizobium cannot fix the nitrogen, these bacterial nodules to another plant. So there is a relation between the plant and the genes. The next one, the nitrogen fixation. The nitrogen fixation. So some bacteria fix nitrogen in some higher plants also. So same account regarding the nitrogen fixing bacteria. There are symbiotic and asymbiotic bacteria. And some bacteria fix nitrogen in some higher plants, that is non-legume plants. So the rhizobium, I told you, the rhizobium with legume plants only. But some other bacteria also fix nitrogen in the roots of other plants, like actinomycetes fix nitrogen in root nodules of Ulmus, Casuarina, Myrica, such plants. And Klebsiella, the bacteria, produces leaf nodules in Pavetta, Diascorea, and Psychotia. In those plants, this Klebsiella bacteria produces the leaf nodules, in the leaf. And actinomycetes bacteria fix root nodules, and these are not this is what's called legume plants, non-legume plants. So nitrogen fixation in non-legume plants. So in the another process by ammonification. Ammonification, so the dead and decay materials are converted into complex proteins into dead bodies of plants and animals, ammonia. So they convert the dead and decay organisms into ammonia, that's called ammonification. So in this, Bacillus vulgaris, Bacillus ramosis, and Bacillus mycoides, etc. convert the dead materials into ammonia. And then the ammonia, already formed by this ammonification, is converted into nitrates by two bacteria. This process, actually the conversion of ammonia into nitrate, is called a nitrification. Conversion of ammonia into nitrate is called a nitrification. So in this, the ammonia is converted into nitrites by nitrosomonas bacteria, and nitrites are converted into nitrates by nitrobacter bacteria. So by the effect of these two functions, the ammonia is converted into nitrate. So plants take nitrogen in the form of nitrates. Okay, then role of bacteria in industries. So first one is the vinegar called acetic acid. This acetic acid is formed by Acetobacter acetobacteria from molasses to acetic acid by reaction of fermentation. So Acetobacter aceti oxidizes the ethyl alcohol, that is from the molasses, into acetic acid by the fermentation process. Next, role of bacteria in the industries. So the same butyric alcohol and acetone, they are produced by the activity of Clostridium acetobutylicum. That is, the bacteria name is Clostridium acetobutylicum, converts this molasses into butyric acid and acetone. Next process is the retting process. So the role of bacteria in the industry. So here the curing of tobacco, tea and coffee. Generally the beginning the tobacco leaves are just green in color, then just they form into light yellow color, they will be removed and stored in very big sheds, and the action of this bacteria, what's called they spray a sort of what's called alcohol. Due to the action of this bacteria, they change their color from green to yellow to brown color you have, that is the tobacco color. The same way tea and coffee also. So the leaves of tea by the Micrococcus conditions and the tobacco by Bacillus megatherium and the beans by the Bacillus bacteria fermented by certain bacteria to import the characteristic flavor and taste and color also. So this is due to the effect of this bacteria only. Import the characteristic flavor, color and taste. Role of bacteria in the industries, that is the retting process. Generally, the fiber yielding plants, they used to cut and put into bundles and they used to keep in water and they keep some weight on the paper, weight on this bundles. After some time, the agriculturist used to take and remove the fiber from the stems. So the process behind this is the fiber is separated from the fiber yielding plants by decomposition of non-cellulosic and cementing material. The process called the retting is the removal of the fiber from the stem by the action of bacteria is called the retting process. The retting of jute, hemp, flax, etc. by the bacteria Clostridium utilicum and Clostridium felsinum. So these two bacteria act upon these fiber yielding plants and help us to remove the fiber from the stems. Next, control petroleum pollution. So generally what happened, it was happen actually in ships and along very big containers etc. There is a leakage in all the oil is spread in the rivers or the oceans. So that what happened, the animals and most of the fishes etc. which found before that they used to die. So that many people try to do it. Like Dr. Ananda Mohan Chakrabarti of India, actually Indian born American scientist, just like Dr. Haragovind Khorana is a native of Orissa and is settled in USA and he conducted many experiments in the pollution and he prepared a new type of strain, a Pseudomonas fetida, that is a genetically engineered strain and this is called super bug. Pseudomonas fetida, genetically engineered strain is known as the super bug and it is developed by him which biodegrade the spilled oil in seas and rivers. So America also given permission to use this technique to clear or degrade the oil in the seas and rivers. Next is the genetic engineering, that is speedily developing subject so that you can get the new characters from one person to another person, even from one plant to another plant, etc. by transfer of genetic materials. Isolation, the transfer of a foreign DNA segment gene into a new host is called genetic engineering or recombinant technology. By this, the plasmid of bacteria acts as the carrier of the gene. Without plasmid, no biotechnology. Without plasmid. So, we cannot take a gene from one person and incorporate it to another person directly. We cannot do it. We can do through the plasmid only. So, many new experiments are done in this and have this regarding the diabetic patients. They used to carry this insulin producing genes are available, they may be incorporated so that we can reduce these things and many hormonal disorders people are given particular hormones through this biotechnology. Thus plasmid of E. coli and agrobacterium tumefaciens have been very much exploited in research work. They are only used here in this research work of genetic engineering. Biogas. You know, gobar gas, biogas, etc. That is nothing but methane. This methanobacillus occurs not only in the rotten vegetables and fruits but also in the dung. It produces methane gas under anaerobic conditions by the fermentation of this bacteria, this methanobacillus, and this is a source of power or source of energy, the biogas. So the biogas from the process of methanobacillus by anaerobic conditions, you can get the gas. And the biological control. Biological control, so we have to control the diseases or pests by using another biological organism and without affecting the plant or animal, that is called biological control. In this, Bacillus thuringiensis infect and kill the caterpillars of some butterflies and related insects. And this BT powder is found in agriculture offices that we can also use. So when this caterpillar eats this powder and crystals are formed in the gullet of these caterpillars and caterpillars die. So this is a biological control, but this animal never affect the host plant. These bacteria do not affect other animals or plants. In the same way, Bacillus papillae kills Japanese beetles and Bacillus thuringiensis kills malarial mosquitoes. So in the same way, the biological organisms which kill other things, that is called a biological control. So by this we finish this bacteria, the long topic, and the very important things in this is the total. All diseases you have to know, the nutrition is important, and the gram stain is very, very important, and biological control, a special topic. Another thing, the control of the petroleum pollutions. Petroleum pollution also you can have. Right? Thank you very much. So tomorrow we'll go to the next group, next topic, algae. Thank you.